Good, so we're online. Reality in images. And for me this is important, and the slides come from Marco Punta, who's now in London, because when we talk about protein structures, they are so visual. I'm going to talk a little bit about energies today, and all of that seems to be so intuitive. But the point I want to make is that sometimes what is very intuitive and very clear may not be as clear, may not be as much reality as we assume. Let's begin with this one here. What is that? What do you see? It's a painting. That was not the question. But what do you see on the painting? What do you see displayed? Anybody?
Trees and huts.
Can you be a little bit louder?
Trees and huts.
Huts.
And trees.
Trees and huts.
Yeah. Skin.
Skin. This is a cool idea. No, I mean, so science works a lot about people who have a very different way of looking at it. I've never heard anybody say skin to this. You're not right, but you are right. Keep following up on associations. This is what's going to get you somewhere. So yes, the title is Georges Braque, and the title is Houses, and yes, that is supposed to be a tree. That, where is that? I already gave it away in the recap. That is a cell, and this in fact is Mycoplasma genitalium, sort of the smallest organism, one of the smallest organisms, and it is completely densely packed. That is a eukaryotic cell, equally densely packed. What is that?
DNA pair.
It could be a DNA pair. It's not. Somebody else?
Oxygen.
Oxygen. Ooh, that would be a lot of molecules. It could be a lot of oxygens. Yes, it could be a bubble of oxygen going up in water. No, not in water. In some liquid where it doesn't dissolve. Lipid.
It doesn't.
It's not oil. It's not oxygen and oil. It is closer to the DNA. Yes, DNA has been said. But it's still not it. It is closer to the DNA. IMAC?
Adenine triphosphate?
No, is not triphosphate. It's closer to DNA. There was something else in between that I didn't hear.
Proteins.
Proteins. Yes, this is in fact one protein that consists of two units, the red and the blue, and every single dot here is a major amino acid. It's not a major amino acid, it's one amino acid. So essentially, in this view, you represent the central C alpha carbon atom that symbolizes an amino acid and the connection of it to its neighbors. This is the same molecule displayed in a slightly different way, where now, instead of just having the single molecules, you may be able to sort of guess that this could be like a cylinder, okay? And this is exactly what we do here. We look at, are there sort of consecutive cylindrical helical structures in there? And I'll talk in this lecture about how this will be done in 2 weeks from now. So how you can, from 3D coordinates, assign such a helix. But essentially all that is done here is you visually highlight some feature, some commonality. Okay? Principle is the same thing. Now, for some, this happens to be a particular protein called alkyl dehydrogenase that some of us in the room have. When you spend too much time at the Oktoberfest inside of the tent and get too much exposure to liquid material that is close to yeast, then you need this molecule to get rid of it, or to sort of feel somehow good when you leave the tent, or next morning, or next day, or whatever. And most likely people in the room who have much less of the activity of the ADH, and therefore they have a problem with digesting the alcohol. It's much, much tougher for them, because that is the thing that breaks it down. This molecule, in fact, again, has the structure here, and like in this picture before, it's more difficult to see here. There's some symmetry. You may recognize that the symmetry axis runs somewhere here, and the molecule up here and the molecule down here, in fact, are two molecules of the same protein, and they have, in order to act and to do their function of breaking up the alcohol, they have to come together. So they work as pairs. And that is also on the other picture that I showed you. What is this? Or what is your association? So let me be very clear here. It's very unlikely that anybody who does not know what it is because they know the painter and have seen this painting will get the right answer. This is very unlikely. With the Georges Braque in the houses, this was much more obvious. Here, it is not. It's more about what is your association with that image?
Sunset.
Sunset. Fire. Cold. What?
Burning coal.
Burning coal. Fire, burning coal.
Flower.
Flower. Ah, yeah, sure. Stick to your way of doing it. Dolphins. No, it's cool. It's really cool. So again, you will be very surprised when you get the answer. And in some sense, let me not, spoil it. Somebody else? There was somebody else. So we have fire, coal, burning, sunset, which all I would relate to energy, high level of energy. We have dolphins, where in fact I assume you mean the jumping dolphins. So this in some sense is aligned with this idea of energy. Anything else? So the answer is, I'm not sure you can see that, Umberto Boccioni, dynamism of a soccer player. So some of you may have just guessed that this is the soccer player, and we're not ready to say it. But the more important part that I mean comes in the next slide. So look at this 2D representation of a soccer player. And whoever likes soccer will possibly name the player, will name the teams, will have some observations. But I challenge you, on the 2D image, on this photograph, you do not have the idea of dynamism. You do not have the idea of fire, of energy, of coal, of jumping dolphins. In fact, you have it here, not here. So what I'm trying to say here is that in this room, at least, nobody guessed that this was obviously a soccer player. I mean, very, very clear. But many people in the room guessed some aspect of what the painter wanted to give out as an idea, what the reason behind that painting is. It is the energy. It's the movement. It's the change. And you got that from that one, and you most likely will not get that as easily. Very few people here would, if I remove the title, very, very few people would pick up the title of dynamism or energy or anything like that. And again, so the important part here is you may find me funny when I'm saying that this may be more real than that, given this title. But my background is that somehow both of those capture different aspects of this title. And this is the same in our molecules. So this is yet another protein, and in this particular case we essentially, like before we had these rods, the long red rods for the helices, now we have them as arrows, as a different type of substructure. Again, it's called secondary structure, we will get back to that. Here you really show in more detail a trace, essentially a line for every single model, and that is showing there are different models here because the protein moves. So essentially different time points of the same molecule, if you want. Now again, both of those capture different aspects. And if I showed you the same view that I showed you in the first slide of the other molecule, where I had the single dots, or the essentially one ball per amino acid, then it would look yet different, and you would actually not recognize those arrows here. And this is the same here. So they're different realities. It is a three-dimensional object. But what it means in terms of its behavior has to do with how we read that object. Yes, we can look at it. And again, I'm going to talk briefly about, we cannot look at it like this, because proteins are too small. They are on the scale of 10 minus 10 or 10 minus 9 meters. They're too small to be seen by a microscope. Plus, they are too dynamic. They move too much. So the only way I can trap them is by, in fact, freezing them into a crystal. Well, that is one of the ways of doing that. And then shooting light X-ray beams, very, very big X-ray beams in CERN, upon them. And from the scattering, I essentially get the structure. But then that gives me for every single atom a 3D coordinate in space. But this is not the reality. It's one reality. Another reality is how that interacts. right? That has to do with the biophysics behind it. And now we get to the constituents, the amino acids. So that is every amino acid has a backbone and a side chain. So backbone means this is the thing that they all have, and the side chains is the feature by which they differ. So these side chains, if I went back to this image of a pearl chain that you throw in solution, protein is a pearl chain with different number of pearls strung up, then the R here would be different colors and different sizes of pearls. Okay? When we connect two of these amino acids and we form a dipeptide, so di is 2, peptide is just this unit, we get this one out through a peptide bond and create the first dipeptide, so a pair of two amino acids where we have essentially two forces. The two protons push each other out of the way, and the electron and the proton attract each other. This is essentially given by such a simple energy function here. So there is an attraction, and then if you get too close, the proteins begin to, same charges, begin to pull each other apart. If you now looked at somehow historically a distribution of the electron cloud, then this is one way of describing it, this is another way of describing it, and this is yet another way of describing it, where essentially you have the protons of this molecule here and the electron cloud around it. And again, it's the same story as before. All of these are approximations. So now we are talking about a very, very, very simple molecule in chemistry, but again, it's the same thing. We have an image, the Bohr's atoms model gives us an image. Typically for chemical processes, this is relatively easy to get from that image to reality. When you add up more particles, this is getting increasingly difficult, and that's exactly the situation of proteins. So 2 is a two-body problem, we can easily solve it somehow. 3, we already know is complicated. Earth, Moon, and Sun, you all know, is difficult to compute. But beyond that, it gets immediately very, very complicated. And as I said, many proteins are over 100 residues long. No way to really easily compute that. Okay, and then you keep going, you string them up, and you essentially join amino acid into proteins. And again, there is the part that is the backbone, and there's the part that is the side chain. When they are joined like this, there's a peptide bond, and they form there are particular angles that can be occupied. Somebody in the last lecture had this idea we could simply compile these angles, and from these angles compute how a protein structure would look. And that in fact is one way in which people try and somehow succeed, but the whole thing is too complex to do that easily and to keep going and just use those features. So let me rationalize. So typically some people may tell you in chemistry there's a molecule, that molecule has a certain feature. They are biomolecules, so we call that a biophysical feature. My point is, this is a rationalization. We cannot quite measure it. We can write an equation that postulates some feature for the whole molecule, but it's not the truth. Again, like in reality and images. It's just one way of describing the truth. And this is the same here. We are rationalizing what we see with simple terms that we can easily understand. So again, this is the structure of some simple amino acids. They again they differ in the side chain they have. Some of these are much longer than others. That is sort of the image of it. And some of these, in fact, have different charges. They have, for instance, different features, such as charges. When we go into the list of 20 amino acids here, these two happen to be positively charged. Okay? There are others that have under some conditions, somehow positive charges. But these are really positively charged. These are two are negatively charged. There are some that sort of can be induced into, so they're polar. They can be induced to form dipoles. So they have, let's call it some potential charge. But together as a team, they have an influence. They're not directly charged, but as a team, they have an influence. And these type of features, another one is a bunch of amino acids hydrophobic. Hydro is water, phobic is be afraid of or against, so they want to be away from water. And then there are others that are friends of water, so-called hydrophilic, because philios is the friend. The Greek philios, not the Latin philios. The Latin philios is the sun. Anyway, whatever. So the hydrophobic, hydrophilic, and hydrophobic stick together. Hydrophilic get to the surface of the molecule and bind to water. And you already have a simple model in which you can imagine how a protein folds. You take the ones that want to be next to water, put them on the surface. The ones that want to be next to each other, put them together. And of course, you have a given change, so you cannot do that with all of them. You have to somehow optimize. That would be the simplest energy, and if it were that simple, we could easily simulate it. On these two features, we can easily simulate a molecule with 100 residues in the solvent. But again, the story is not that simple. Now there are subregions, and in this one, I present a level of complexity of biology that, again, I do not really expect you to understand and swallow, but I want to have it at least passed in front of your face. So that's the RNA, or essentially the DNA becomes RNA, and the gene begins somewhere here and ends somewhere here. And in this gene, you have you observe, let's call it blue and red. Happens to be exon, happens to be called intron. So what happens now in the next step is that the blue parts are taken out, are simply cut out. The biological word for that is spliced out. Only the red part here are put together, in this particular case, those are 3, and form the protein. A domain, there's a typo here, a domain is a subregion of a protein that is relevant for function. So early on, there was a suspicion that maybe the domains relate to these red regions here. So that in this particular case that I show, you essentially would have a protein with 3 domains. And there are publications to the day that claim that there is some truth to this. Largely, arguably, this is not very close to the reality. There are some proteins where you can see that. For the majority of those, this doesn't hold up. But anyway, so there is this structure in which you simply cut out part of the gene, and this red part is the only one that will become the eventual protein. It is assumed that the blue here and the green are all important to contain the important information for how the red one is expressed, meaning how many of these red ones do you get as a copy, how early it is done, when it's done. So all kinds of regulatory signals. That's supposed to be in the green, before the gene, before the green, and also in the blue one, sorry, the one that is cut out. So they are supposed to be important. Now, again, on the human genome, when I look at the entire genome, that is 3 megabases, so we have 3 billion nucleotides. And we assume that what is red here makes up only 1% of these 3 billion. Okay, all the other parts are green, before the green, and blue. So in some sense, in terms of operation, thrown away. For many, many years, people believed they are junk. Today, the belief is more that they have some importance for regulatory functions. But how much, we still don't have any clue. Okay, let's get back to the evolution, to this issue about how proteins evolve. There are situations where you have one particular organism and a protein that is indicated by this bar here, protein A. And in the same protein, you may have a protein B, purple here. Now you may have another organism in which you have one protein C that merges these two. Okay. Here's another way of looking at domains, and now this is really the same image as before, but showing you the structure. And you may or may not see that there is some similarity between this and that. So this one now interacts. It's a similar situation as I showed you before. So you have something that is a single molecule here in one organism, and is joined with another one in another organism. And it's still the same sort of unit. Christine Orengo is talking about embellishments. So this is another kind of that story, where you have a smaller thing that is repeated in the larger thing. This figure here shows this on a larger scale, where you essentially pick out a certain theme here, and that theme would then be one yellow triangle. And you ask, how often do I find the same theme in other proteins? And you do see that there is a bunch of proteins here, and that they have a bunch of these themes or motifs or regions that come in different combinations, but repeatedly. So many of them have this yellow region here, many of them have this purple circle here, but the way they are aggregated, or they're strung together or put together, this is now no longer a string, because they essentially hold together in 3D, is slightly different. Although there are commonalities, as you see, they always start with the circle and so forth and so on. This is essentially one way of explaining subunits of proteins, typically related to, referred to as domains. Yeah?
What is the meaning of these yellow arrows?
So these yellow arrows, and I'm sorry for that again. So there are two, I'll explain that again. But there are two major substructures in a protein. They're shown up there in purple and yellow. One is this helix that I showed before as the rods. And the yellow one, the arrows, are so-called strands. And those are the sort of major, they're called alpha helix because it was observed the first. Again, I'll get back into these words later. And beta strands. Alpha, beta, the first two. And in fact, those are the main elements. The reason for showing an arrow is to simply indicate in which direction does the protein move. So it moves, that's the first residue in that strand, and that's the last residue that goes in that direction. And you sometimes have parallel arrows here, so you have parts that go in the same direction, and you sometimes have anti-parallel. And they are very, very important for the recognition of units or for the formation of units. So again, the so-called secondary structure shown here as a word. We'll get back into that. But the major point here is there are units. We call them domain. One idea about a domain is, and this is not quite true, one simple image of a domain is something that folds independently. So you take that thing, you throw it in a solvent, you get always the same three-dimensional structure. Although I said this about a full protein. But it may also happen with subfragments of the protein, while if you cut out other fragments, like you just cut out this part here, it will not adopt the same unique three-dimensional structure. It will just be a wobble. Okay? But there may be regions that are sub the entire protein that have exactly the same features as the entire thing. The feature being, you take it, you throw it into solvent, you always get the same 3D structure. And those we tend to imagine when we use the word domains. Sorry for being so vague. The way we guess domains from sequence, and now I introduce something that you will see again and again and again. And you already have seen it. So the bar simply indicates one protein sequence. Okay? And when I put another bar in here, protein A and protein B, what I mean is that protein B has some similarity to A in its sequence in that region. So there's a part of A that is not similar to B, and there's a part of B that is not similar to A, and there's a part where they're both similar. And you keep going for another bunch of proteins, and looking at something like that, you can possibly guess that this, in fact, here would be one domain, another region. Because it is shared, there's something common shared throughout many organisms. And then the suspicion is this could be a domain. It could also be, if it's too short, it may just be a binding site. So it's something, binding sites are tens of residues, 10 or a few to 10 residues, or 20, 30 residues, while domains tend to be more 100 residues long, okay? When we look, and the important part here is the blue, when we look at all the proteins and ask how many proteins have a single domain, then the answer is in blue here roughly 30%. There are actually more proteins with 2 domains. And then you see it continues. So most proteins really have more than 1 domain. Therefore it's true that longer proteins are rarer than short ones, and that most proteins are indeed multi-domain proteins. Let's not get into too many of these details, but this is important. This I should have cut out, I'm sorry. Most proteins have more than 1 domain, that's important. And these, I'm sorry, this is too detailed. Okay, let's get back to the principal idea of 3D comparisons. And what we want to do is we want to look at 2 proteins and answer the question whether or not they're similar. Okay? Let's just begin with a totally simple idea, so of matching shapes. In the simplest idea, you take the circle, you overlay it to the square and the circle, and you simply decide, okay, one is more similar than the other. And the way you decide that is somehow by overlapping it, cutting out what is left over, right? In this particular case, you see that the circle fits much better here than here, because there's sort of a leftover. And the other one there is not. Then there are somehow leftovers that come from slight deviations like that. But in order to do that, what you have to do is you have to find corresponding points. That means you have to first find the orientation in which they fit the best. Going back into the example that I had before, if you were to just move the circle over here, or this one over here, and that one as it is shown, then there's less left over here than there, right? So you also have first to find the best possible superposition. There's a difference in corresponding points. In this particular case is the case, so you find the best position. In order to find the best position by an algorithm that is doable, easily doable in a time that we can use for standard comparisons on a standard computer, we in fact have to get to the point of points. And the points here in biology would simply be the amino acids. So each amino acid is a point, and we essentially align or find the corresponding points as quickly as possible, and then we simply optimize the way of superposing them. And the simplest way of superposing them is to look at the root mean square deviation, essentially where for every single point between the 2 objects, we compile the Euclidean distance. Okay? Clear? Any issue with this? It's essentially a vector difference, yeah?
Why do you have root domain? What's important about this?
Why do we use your root mean square deviation?
No, why we use domains?
Why we use domains?
Yeah.
So let's make this clear. So there are 2 different issues here. My first statement was, so there is a molecular unit that is an atom. The atom leads to a molecule, and one particular type of molecular amino acids. These amino acids are the units of proteins. Another unit of protein is sort of a structural unit. It's something that, if I look at many structures, I recognize the same thing again and again. And that, for instance, is a secondary structure. We get back to that. But another unit that is recognizable is the idea of a domain. So we need domains in order to identify similarities in proteins. That's one way. For me, as the computer scientist who wants to compare structures, I need domains because they help me to, in fact, find the right parts. So to decide, I'll get back to that in a minute, which part is the circle, in some sense. And this circle here corresponds to a domain. Why do I need domains on a level of evolution? Is essentially, imagine them like building blocks. And by having smaller building blocks, you can easily move around in the space of evolutionary possibilities, meaning you could just copy one domain, and as I showed you in this example where 2 proteins interact, that you possibly could copy two domains as one protein in the next organism in order to increase the probability of interaction. So there, the answer to your question, why do we need domains? To make a job faster would be then the answer. Another answer would be because it helps evolution, because it helps the exchange of material. Of course, this is not the answer, but it's the other way around. They have evolved because they are very fit to evolve. So we are getting into this circular argument. It's a longer argument, I'm getting too deep here. But essentially, the domains are useful in a biological context, because they allow easy replication and allow, over the course of a couple of billion years, to create new things easily. It's like Lego blocks.
But then we're not sure. Like, we have amino acids that can be formed with different sequences. So if we concentrate on structure that can be different for the same amino acid, but that have been formed with the different sequences. So how can we predict that this domain fits every instance of time?
So how can we? Let me try to repeat your question for the camera and for the others. The way I assume you say that is we know proteins consist of different amino acids. How can we predict that two sequences with different amino acids fit into the same domain? And the answer to your question, unfortunately, I'm going to do this sort of thing that is not nice, but the answer ultimately is the lecture. There are a couple of ways in which we can do it and not, in which we cannot do it. What I'm trying to do today here, in this lecture, is I assume that somebody gave me the 3D coordinates of every single point. So somebody made the experiment. And now what I want to do is I want to, first of all, gaze domains, so just look at it and see subunits that form domains. And I want to compare two of these three-dimensional objects. And that will be it for today. Your question will take much longer to answer. So this, in fact, is exactly the challenge. We cannot predict from sequence 3D structure in general, so therefore we can also not predict domains. So the short answer is we cannot. For some cases we can, and those are the cases that I will talk about. Anything else? So again, we are in this image of RMSD, or in RMSD describes the similarity between two three-dimensional objects given points that I have assigned. So it's intrinsically not a continuous measure in that sense. And in these particular cases here, visually it would appear that the RMSD for this one here, for these two, for the superposition of these two objects is much smaller than the RMSD for these two. Now, in order to do a 3D comparison, first again we have to find the corresponding points and then superimpose and then do the RMSD. Now what about this one here? In this particular case, we would have to do it like this, fairly easy. Now let's go back to the proteins. The question is, how do we compare them? Do we compare them as entire objects or not? So as entire objects, in this particular case here, let's go into this example here. Let's call it global versus local. Where global is the idea I treat them as entire objects. Local, I somehow cut them apart into something that I call domain, or whatever I call that thing I cut them into. So let's look at this object and that object. So the question is green and red, are green and red similar? And the best way to do that is, in fact, so here are different global solutions for superposing them, and they are all more or less impressive. But if you cut up these objects, then you see that you can easily fit these two to essentially all pieces fit to each other. Every piece in the green you can fit to a piece in the red, and by that token you could say that they're completely contained in each other. Now, of course, this depends on how you cut. In the most extreme example, you cut it into a line, so the green object has lines, and if I asked how many of the, essentially every single line, is the line found in the red? Sure. every line is found in every object, right? So the cut down level line or point is irrelevant to answer the question whether the green and the red are related in any way. All points are similar, particularly all mathematical points. So we need to find the valid unit for comparison. How can we find the valid unit for comparison? So this trivial example, points or lines are not it. Any idea how we could decide what is a valid unit? Anyone?
We can use coordinates.
Can you try to speak up a little bit?
We can use coordinate system for predicting the position of.
So we can use a coordinate system, but how would I use a coordinate system to predict what is a relevant sort of cut? So again, the question is, on this example that I showed before here, the single line is too much of a cut. Yes, this will be found in the red, but it will be found in any other object I can draw. I can easily draw an object that has absolutely no reasonable similarity to this green thing here. And yet we'll have a line. So thereby I would argue the line is too much. So cutting it down to the level of a line is not reasonable as a unit. But cutting, I mean, again, visually, we all very happy to immediately say, okay, cutting it to a circle, to a rectangle, to some triangles, this, for that object, or getting back to the comparison between the red and the green, this is reasonable. But how do we find the reasonable parts? How do we distinguish between line and these objects that, again, visually you immediately see which ones those would be? Sorry?
The objects there are also made of lines.
Yes.
But the granularity of those lines is much, much finer than the unit of line which we use to cut the object.
Yes, so that is exactly the point. Let me repeat. So they are all made of lines. But again, my point is you could draw an object that is completely different, where you have absolutely no similarity to the green or to the red, that is also made of lines. So how can we, just a sec, how can we find the distinction between something that's totally unreasonable, line, it is unreasonable because it captures anything you can draw, and something where you feel I can break it down into sort of macroscopic subunits, so not points, not lines, but something that is reasonable to establish there's a similarity. Yeah?
Maybe we should search for two so similar proteins that they differ only for a very small part, and take it for one of these figures.
So the idea here is, let me illustrate that idea, and send you on one of the graphs I had before. Find two proteins that are very, very similar, such as this sort of whatever color is, and the green one here.
And we get this light green, how to say, area for the part that we distinguish.
Exactly. Oh, that's an interesting idea. So let me repeat to everybody else. So what he's saying is that if I found an object or a pair of objects like this, then the part, this half moon here, the cutout part, is exactly what should be the reasonable unit of cut that I should begin with to compare objects. This is a great idea. The problem with that idea is that if you found an example like this, and you believed that the similarity between two parts of this, so essentially for the green I would have two parts. I would have the moon part and the circle part. For the circle part, I would know there's another protein that is exactly the circle part. For the moon part, I may now go out and find, is there any other protein that is like the moon part? And I believe that would be a great idea. Now if you didn't find any that is like the moon part, then you are left with something that you never find again, and something that you find again. So the circle you find again, and the moon you don't. So then you would actually argue, your idea is a good one, I would just invert it, And you would argue, in this particular case, the unit then should be the circle. So by a comparison like that, I would find out that in fact I should look at circles. And I believe this is a great idea. And in principle, this is the way people do it. The problem a little bit, but again, this is the way science works. The problem, of course, is sort of circular. You want to find the unit in order to compare two proteins. Because you don't know the unit at the first step, you compare two proteins. Hello, how can you do that? But essentially you begin with the entire thing, you see, do you find anything like that? And then you call those units and you do a refined step. And this is exactly the way it works. So now my animations are in my way. And this one is saying it in a slightly more formal way using, let's call it statistic or heuristics, where in fact you have somehow background. So you look at something, it's a similar idea that you bring, but you look at some units and you ask, what's the probability of matching it anywhere? So the line you would match to any object, so it's not very informative, you would argue, while the circle, you would only find it some proteins, so you would somehow have a signal that stands out from the random background distribution. And that's another way of doing it. And this idea we get back into. Now this idea, again, you have something that you possibly could do by randomly creating things, or by looking at all the proteins. And we do know that all proteins somehow are related. Life evolved from some RNA molecule. In that sense, proteins are evolutionarily related. But we also believe that protein structures are very different, and that the particular shape of proteins relates to the particular function, and they are not all the same. So meaning that if I just took all the structures that I know, and I took that as the blue, as the background, then for most, I do not want to match the pieces that I call important units. I may match to many, but not to all, or I will not match to all, to most. Is a sort of simple statement? Is that clear, what I'm saying? It's simple but not easy to digest. Okay, I assume yes. So again, you cut, you match, and you compare. One way in which you can do that is just to map it onto a matrix. You compare, in fact, so essentially you have a matrix where you put, in this particular case, we have 8 points, and maybe there's another one where object that has only 7 points in red here. So we compare an object with 7 points to an object with 8 points, and then every single point here, we ask what is the proximity of those to each other. We essentially translate the three-dimensional object into a two-dimensional objects of contact. And then through this two-dimensional objects, what we then get is we have two-dimensional objects, and we can simply compare two matrices. That's another way in which we can do the 3D comparison. Okay? When I take a three-dimensional object and I project it of whatever it is, in this particular case here I have dots on a 2D landscape. If I took dots on a 2D landscape and I drew the matrix of the proximity of all the dots in one object, and I compared that to a three-dimensional object. So I take the three-dimensional object, whatever it is, a ball or some shape, some complex shape. And this complex shape, I have points. And now I take these points, use them to compile the distance of any pair of points in this object. My question to you is the following. Is there anything in this three-dimensional structure that is not contained in the 2D matrix? Are these 100% equal maps in terms of an information content of each other? So if I only gave you the entire distance matrix, could you reproduce the 3D object? Again, what I'm assuming is that the 3D object is defined by points. I'm not talking about I just use the word sphere, it's a little bit misleading. I'm talking about a 3D object made up of points. In the examples that I show, those were 8 points. Let's just start there. I put 8 points into 3D, compute all the pairwise distances. I have a matrix of 8 by the 8, and I'm asking the matrix of 8 by 8, is that the same information as the full set of coordinates? I hear a lot of no's. I haven't heard any yes yet. Anybody who is saying no, what is missing? He says depths, and down here somebody who says connections. Wait a minute. The 2D matrix has all the connections. Or do you mean connections as in 3D? Well, what is a connection? A connection is proximity in space. So if you put if point 1 and 2 were next to each other, then that's what you would see in the matrix. In fact, if 1 and 2 were the most similar of all the 8, of all the 8 times 4, 32? No, getting that wrong again. 4 times 28. The diagonal is trivial, right? So of all the 28 points, then I would immediately see that because it's the minimal value in the entire matrix. So connections I have. Depths. What do you mean by depth? What is depth?
I mean, the 3D structure, so every point is also like that. So
Yeah. Every point has a position in 3D. And you assume that you lose that.
Yes.
Now I challenge you, you don't. So the only answer that I heard was no to my previous question. And the answer is right. But it is more subtle than you believe. The only thing that you cannot distinguish is the three-dimensional object and its mirror. Everything else you can do. And there's a clear algorithm. Depth you can do. It's fully in this matrix.
So where is it encoded?
In all the pairs. You essentially can find the optimal path through all the pairs and it will reconstruct the 3D space. The only problem in this algorithm is you cannot distinguish between this and that. So between the original and its mirror. That's the only piece of information. Therefore the answer no was right. You do not have the same information, but it's actually only one bit that is different. The depth, this is sort of what you believe, now what we see as the most important part of the 3D information, is actually available in the 2D matrix. And that ultimately is the reason because the 28 numbers that I'm talking about here contain a lot of information. And in this, a lot of information, you actually store the entire 3D matrix. That's an important reality when you handle 3D objects, because very often it's much easier to compare 2D matrices than to compare 3D objects. What I talked about when I said RMSD is the assumption that I compare 3D objects, and I only use my circles and squares because it's easier to show that. But in principle, all I said so far was valid for comparing 3D objects. While now I translate the 3D object, put it into a matrix, and I do a different way of comparing them. Okay? And this is in fact exactly what is done in biology. So some of these slides are from Patrice Coe. So again, in this particular example here, now assume I wanted to do an alignment to itself. Then in order to see similarities, you would have to somehow break it up and then compile the RMSD. In this particular case, for instance, you see there's some similarity of this region to that, and there's some similarity of this region to that. This is the overall similarity. So C simply means that's main atom, so one per amino acid, main carbon. And this one is 2.85 angstrom. Angstrom is 10 minus 10 meter. So it's 28 nanometer or something, whatever. 2.8. 2.8. Which one is better? Those are two different alignments of two different parts. Important is not, so the answer to my question is not clear at all, but what is important to remember is the fact that A is similar to B and that B is similar to C does not mean any similarity between C and A, because proteins consist of units. So now back to the question, why do we have domains, or why do we have units? So they really get into our face in this particular example. So they are really making it very complicated. So I'm not answering why do we have domains, but here I'm saying domains are a curse. I wish we wouldn't have them for this kind of situation. That immediately kills, for instance, the metric idea. One concept here from Taylor and Arango is a method that essentially looks at all the pairwise similarities in certain local regions and then compares two local matrices, not the entire matrix. So here you do have a loss of information in this particular example. In fact, the loss of information here has to do ultimately with the depth, because you're looking at only small regions. And then you lose indeed the depth if you cut out from the distance matrix something around the diagonal. Then you exactly lose the depth. But it's a fast algorithm, and you simply find the optimal way to compare these two matrices. That's one way. The problem is the loss of direction, of depth of important information. Here's another one that uses, and that's what I said before. So essentially you're looking at a sequence here. The sequence is 1D, is a string. That's the 3D structure. You have the three-dimensional information. And then they have a 2D matrix where you essentially write every single residue, every single amino acid here. Let me just get back to that notion, and I will confuse these words again and again over the course of my lecture. When amino acids, so that 20 proteins are made up of amino acids, there are 20 different amino acids, they're the units of proteins. When they are put together in a protein, we typically refer to residues. So residue is one position in a protein, one letter. Okay, so essentially what you write down here is the 98 residues in that direction, 98 residues in that direction, and here is essentially the darker, the closer. And that's a distance matrix. So again, from the 2D representation, you can essentially get back the 3D representation, but in 1D you have none of that information left. And this method DALI, in fact, now does takes the full 2D matrix and finds in Monte Carlo that somehow of a hexapeptide, so moves hexapeptides around, so large groups, 10 or something in that ballpark, and compares them in the other one and finds the best path to make these all fit. There's another one here, Voroline. It's a 3D alignment that in fact comes from the LMU. And where you essentially describe every single position by its Voronoi environment. And then you simply go out and see in another protein, do you have a similar Voronoi environment? And then you build up the alignment. Okay? The major thing here always is this issue, we cannot do full proteins against full protein, we have to somehow chop them up. Here's an extreme example. The two forms of the calcium bound chimodulin. Chimodulin is just one particular protein. This is one, and this is the other. It's the same protein, but it looks very different. So if you took the red and overlapped it with the green, there's absolutely no similarity. But if you cut it up, in the global alignment here, you have a huge RMSD of 15. In a local alignment, so where you cut it up into the pieces, you see they're, of course, they're identical. There are many, many other methods to compare, but this part was only to give you an idea that it is important to compare these three-dimensional objects. There's a whole school of methods out there. I'm not going to go into that. What we will go into in more detail next week is how you compare sequences. And from there we will get into predicting protein structures and into the question how we can see whether two sequences that are different have a similar domain or not. Again, cells are densely packed, there's an inside and outside. Proteins thrown into solvent adopt a unique three-dimensional structure. These structures come in very different sizes. Here's an example of the outside. This is it's a slide from Natasha Wood. On the cell surface, you have essentially receptors standing out. Those are images of those receptors done on the base of the actual experimental three-dimensional structure. That's a glycoprotein 120 that comes from HIV. And in fact, infects this cell here by docking to these receptors. That's that image. Here is a little more detail, and in this little more detail, the point I want to point out is whether or not that docking happens has to do with a region in the protein here that is referred to as a loop. You can see it as a loop. It's like a lasso loop or something like that. And the way this is shaped, the way this is mobile, allows it to enter or not. This is why this is one of the drug targets. We're trying to change this loop, or not we, they are trying to change this sort of loop in order to make the HIV glycoprotein no longer be able to intrude into the cell, the so-called V3 loop. There are 3D modules, domains is the word I use, that are repeated. Those are important units that we need to compare proteins and that we in fact try to predict from sequence. Okay, we get to next Tuesday where we compare proteins from the sequence, and this process is called alignment, and I'm ready for questions.